Welcome back to the Science Experience, where we explore the most exciting new research papers and preprints just for you. Before we dive into today's fascinating topic, could you do us a huge favor? Please hit that like button and subscribe to the channel. It genuinely helps support the Science Experience community, and, it lets us bring you more insights just like this one. Okay, so today we're embarking on a deep dive into a molecule that has, for a long time, carried, let's say, quite a bad reputation. Reactive Oxygen Species, or ROS. For decades, the general consensus has been, that ROS are simply damaging byproducts of metabolism. Cellular villains causing oxidative stress, driving aging, and all those age-related diseases we really want to avoid.
Right, but what if that's only maybe a small part of the story? What if, under very specific conditions, originating from precise locations within our cells, some ROS aren't just harmless, but are actually vital messengers? Molecules that actively promote health, enhance resilience, and maybe even extend lifespan. That's the surprising, evolving narrative we're going to unpack today.
Right.
And honestly, it might profoundly change how you think about these often misunderstood molecules.
Okay, let's unpack this reversal. It sounds pretty significant. Our journey into this new understanding, it really began with some early, quite unexpected findings. Findings that started to, poke holes in that traditional villainous view of ROS. Groundbreaking work in simpler organisms like the humble C. elegans worm showed something truly counterintuitive. It showed that carefully increasing certain mitochondrial ROS could actually prolong their lifespan. That's a real head-scratcher when you've always been told ROS equals bad.
It absolutely is. And what's fascinating here is how researchers started to pinpoint the source of these seemingly beneficial ROS. It quickly became clear that the site of production within the mitochondria profoundly influenced their effects. This particular line of research zeroed in on a specific part of the cell's energy-making machinery, known as complex 1 of the electron transport chain. And crucially, they identified a unique process called reverse electron transport, or RET. You can sort of think of RET as electrons flowing backwards through this part of the energy system. And in doing so, generating a specific burst of these ROS.
Right, backwards flow. So that's where it starts to get really interesting. They found that by carefully manipulating the mitochondria in Drosophila fruit flies, for instance, by introducing a yeast enzyme called NDI1, they could boost ROS specifically via this reverse electron transport through complex 1. And the striking result...
Longer life.
Exactly. These flies lived longer. It wasn't just a small bump either. It was a significant, measurable extension of their lifespan.
Yeah, this really cemented the idea that these RET-derived ROS were acting as a powerful, positive signal, not just random damage. They showed that this beneficial signaling depended on what they call the overreduction of the Coenzyme Q pool. You could imagine the cell's energy shuttle getting really full or highly charged, and also a sufficiently high membrane potential, like a cellular battery needing to be fully powered up and ready to fire off that specific signal.
Okay, so those conditions have to be met.
Precisely. And what's truly remarkable is how powerful this signal proved to be. It could even rescue mitochondrial dysfunction and extend lifespan in flies that already had severe oxidative stress, or, those mimicking age-related declines in critical proteins like PA and K1. It just completely shifts our perspective, doesn't it? Showing that the source of the ROS determines its physiological outcome. We're moving beyond the simple ROS is bad idea to something much more nuanced.
Definitely more nuanced. So, we've discovered this specific complex 1-derived ROS signal can actually extend lifespan, at least in model organisms. But that raises an immediate question for me. Is this just something we see in the lab, or does our body naturally use this surprising mechanism, to adapt to everyday pressures? Does it play a role in our regular physiological life, especially when we're under stress?
That's a crucial next step in the research, and you've hit on it. The answer seems to be a resounding yes. Researchers demonstrated that heat stress in Drosophila flies, for example, actually triggered a natural physiological increase in mitochondrial ROS in the brain after just a few hours. And critically, they confirmed this increase was indeed due to reverse electron transport. So it's not just some lab trick. It seems to be a built-in natural response.
Wow. Okay, but how did they manage to verify it was specifically RET-derived ROS in a living organism? That sounds like a pretty tricky thing to prove, right, given everything else happening inside a cell.
It is tricky,
right,
but they used very specific molecular tools. For instance, they used rotenone. Rotenone essentially acts like a roadblock, preventing electron flow backward through complex 1.
Right.
Or they use something called FCCP, which disrupts that crucial membrane potential we talked about, that battery charge necessary for RET to even happen.
Right.
And both treatments, rotenone and FCCP, prevented the stress-induced ROS increase, which strongly supports the idea that rotenone was the precise source of this particular ROS signal under stress. These tools really let them isolate and confirm the RET pathway was responsible.
Right, that makes sense. They could block it and see the effect disappear. So what happens if you do block this vital ROS signaling during stress? Does it just, well, make things a bit worse, or is it absolutely critical for survival?
It seems absolutely critical. If you prevent this ROS read signal from forming, for example, they did this by expressing an alternative oxidase called AOX. Now, AOX isn't normally found in flies or humans, but it effectively diverts electrons and prevents that coenzyme Q pool from getting over-reduced, which is needed for RET.
Ah, so bypasses the RET trigger point.
Exactly. And when they did that, the flies dramatically lost their ability to adapt and survive under both moderate and severe heat stress. Their survival was severely shortened. It really underscores the critical protective role of this signal in everyday physiological challenges. It's not just a minor player.
That's amazing. So this specific ROS signal is vital for stress adaptation. But when we talk about ROS, that's still a bit of a broad category, isn't it? What exactly is the specific messenger molecule here? Was it like superoxide or something else entirely acting as the crucial signal?
That's another great question. The studies managed to precisely identify hydrogen peroxide, H2O2, as the crucial messenger ROS in this context.
Hydrogen peroxide. Okay. How did they pin it down to that specifically?
Well, they did something quite clever. When they overexpressed mitochondrial catalase, or mtCAT, that's an enzyme that specifically breaks down H2O2, it drastically shortened the fly's survival under stress. This effect phenocopied or perfectly mimicked the effects of blocking RET directly with rotenone or FCCP.
Got it. So removing H2O2 had the same bad outcome as stopping RET.
Exactly. And conversely, when they overexpress SO2, that's an enzyme that converts a different ROS, superoxide, into H2O2, it didn't harm the flies. In fact, it even modestly improved survival under stress.
Oh, interesting. So more H2O2 in this context was potentially good.
Right. It highlights a really fundamental point. Specific ROS, particularly H2O2, generated via RET, seem essential for activating these adaptive mechanisms and ensuring survival under various stressors, heat, low oxygen hypoxia, even high oxygen hyperoxia. This really advises against those sort of blanket antioxidant strategies that just try to suppress all mitochondrial H2O2 signaling, because clearly context and specificity are absolutely key here. You might be shutting down something important.
That completely changes the game for how we think about antioxidants, doesn't it? Okay, so we've gone from this initial unexpected discovery that certain ROS can extend lifespan to their vital, very nuanced role in helping organisms adapt to everyday stress. This is truly groundbreaking stuff. But what happens to this crucial mechanism as organisms get older? We generally know that overall mitochondrial ROS levels typically increase with age. So is it just the same beneficial signal, but maybe it's constantly active in older individuals, perhaps to a detrimental degree?
That's the critical distinction, and it's a bit counterintuitive again. While total mitochondrial ROS do tend to accumulate with age, especially in the brains of old flies, the beneficial signaling-specific ROS RET mechanism is progressively lost.
Lost, even though total ROS are higher.
Yes. Old flies produce consistently high levels of ROS, but these are not the same finely tuned RET-derived signals that aid stress adaptation in younger individuals. They're more like background noise. A general accumulation of cellular static or damage, lacking the specific message and the precise timing we see in younger, healthy mitochondria responding to stress.
So the body loses its ability to actually activate this helpful signaling pathway in response to stress, even though the overall ROS levels are already elevated in older individuals. That's quite a twist, like having a lot of static but no clear radio signal when you need one.
That's a great analogy, exactly. When researchers put old flies under thermal stress, their ROS levels didn't change in that adaptive way that young flies' levels did. There was no specific increase in the beneficial signal. And crucially, applying rotenone, which as we discussed specifically blocks RET, had no effect on these already high ROS levels in the old flies.
Proving the high ROS weren't coming from RET anymore.
Precisely. It confirmed that the ROS being produced were no longer RET-derived. And this loss of signaling capacity severely impacts the old fly's ability to adapt to stress. It significantly shortens their survival when they face challenges the younger flies could handle.
This raises a really important question then. Why does this beneficial ROS signaling disappear with age, even as the overall ROS levels are going up? What's the underlying mechanism causing this crucial loss of function? Is it complex 1 just wearing out?
Well, the research points towards a problem not necessarily with electrons entering the electron transport chain at complex 1, but rather with impaired electron exit further down the line. Specifically, when complex 4, another crucial part of the mitochondrial energy machinery, near the end of the chain was genetically depleted or acutely inhibited, it beautifully mimicked the mitochondrial profile seen in old individuals.
Complex 4, okay. So a blockage at the end.
Kind of, yeah. You saw overall high mitochondrial ROS levels, but crucially, no ability to generate that beneficial ROS-RET signal in response to thermal stress, just like in the old flies. It suggests a kind of traffic jam of electrons. A bottleneck at the end of the chain leads to this continuous, less specific ROS production spilling out rather than the finely tuned signal-generating flow you see in younger, healthier mitochondria when ROS is activated. It's a shift from a controlled, beneficial signal triggered when needed to an uncontrolled, possibly damaging byproduct because the system is clogged up.
Wow. What a remarkable journey we've taken through the complex world of mitochondrial biology. From that initial unexpected discovery that certain reactive oxygen species can actually extend lifespan,
Right,
to their vital, nuanced role in stress adaptation, and then finally understanding the unfortunate loss of this specific signaling pathway during aging. It's a truly profound deep dive. This completely overturns so much of the old thinking about ROS. It's really a lot to consider for our understanding of health and aging.
It absolutely is. This deep dive profoundly changes how we need to understand ROS. It really challenges us to move beyond those simple good or bad labels and appreciate the precise, context-dependent roles these molecules play within our cells. And if we connect this to the bigger picture, it also suggests that maybe future therapeutic interventions for aging and age-related diseases might need to be highly targeted. Not just focusing on eliminating all ROS indiscriminately with general antioxidants perhaps, but maybe on restoring or enhancing these crucial, beneficial signaling pathways like ROS-RET to improve stress resilience and extend health span. What stands out most to you from all this?
A truly provocative thought to leave everyone with indeed. Thank you so much for joining us for this deep dive here on the Sand Science Experience. If you've enjoyed this fascinating exploration of cutting-edge science, please consider liking this video and subscribing to the channel. Your support genuinely helps us continue bringing these insights directly to you right here at the Sand Science Experience.
Until next time, keep learning and stay curious.